The COVID-19 pandemic made mRNA vaccine a household name. We all heard about it constantly. But here's the thing, that's really only half the story. It turns out there's another, older genetic vaccine technology that's just as revolutionary. So let's just dive right in. We've all heard of mRNA vaccines, right? But what about their older, maybe more stable cousin, the DNA vaccine? What's the real difference between them, and why do we even have both in the first place? Okay, so to get this, we first need to wrap our heads around the brilliant idea that connects them, the genetic vaccine. See, instead of injecting a weakened or dead piece of a virus, a genetic vaccine gives our own cells the instructions to build a tiny, harmless piece of it. This piece is called an antigen. And just like that, our body becomes its own little vaccine factory. It's an incredible concept. So if they both do that, what's the difference? Well, the crucial difference is how they deliver those instructions. And that brings us to this great analogy that really makes it all click, thinking of one as a blueprint and the other as an instruction manual. Here's exactly how that analogy plays out. Think of DNA as the master blueprint. It's the original, detailed plan. It has to be delivered to the cell's head office, the nucleus, to get processed first. RNA, on the other hand, well, that's the ready-to-use instruction manual. It's a direct photocopy delivered right to the cell's factory floor, the cytoplasm, where all the protein-building machinery is waiting to get to work. Let's follow the journey for a DNA vaccine. You can see it's a multi-step process. The DNA has to be delivered, it's got to get inside the cell, and then it has to make that critical journey all the way into the nucleus. Only when it's in there can it be transcribed into mRNA, which then leaves the nucleus to be translated into the final protein. Now, let's compare that to the RNA vaccine pathway. See how much simpler this is? The RNA gets delivered and goes straight into the cytoplasm. It completely bypasses the nucleus. There's no transcription step needed at all. The cell's machinery can start reading those instructions and building the protein immediately. It's way more direct. This table really pulls it all together, side by side. You've got different materials, different cellular targets. But this last point on genomic safety is absolutely key. Because RNA bypasses the nucleus entirely, sources confirm there's no risk of it getting mixed up with our own DNA. For DNA vaccines, that risk is considered incredibly small, but because it enters the nucleus, it theoretically exists. All right, so we've covered how they work inside our bodies, but what about how they're actually made in a lab? That brings us to the production line. And this is another fascinating contrast. DNA vaccine manufacturing is a biological process. Scientists basically use bacteria as tiny factories, growing them in huge vats to crank out billions of copies of the DNA blueprint. RNA manufacturing, though, is totally different. It's a synthetic, cell-free process. It's more like rapidly printing the instruction manuals using enzymes in a test tube. No living cells required. So you've got different mechanisms, different ways of making them, and that, of course, leads to very different strengths and weaknesses. So it's time for a little performance review, where we look at the key trade-offs. For DNA vaccines, the pros are huge, especially on the logistics side. First off, they're incredibly stable. Many of them can be stored at room temperature, which is a game changer. Plus, production is highly scalable using that well-established bacterial process. The main con, though, is that they've historically been a bit less potent in humans. They sometimes need a bigger dose or even special delivery methods, like a little electric pulse called electroporation, just to help push that blueprint into the cell's nucleus. Now for RNA vaccines, and it's pretty much the inverse story. Their massive pro is sheer efficiency and speed. Because they act directly in the cytoplasm, they trigger a really strong and rapid immune response. But their major con? Instability. That RNA molecule is just incredibly fragile. It's why it needs to be wrapped in that protective bubble of lipid nanoparticles, or LNPs, and why it needs that famous ultra-cold storage. Okay, so that's all the theory. But where do these amazing technologies actually stand out there in the real world? Let's take a look at these vaccines in action. No big surprise here, right? For human RNA vaccines, the list is obviously led by the stunningly successful COVID-19 vaccines from Moderna and Pfizer. But the platform is also approved for RSV and is already in trials for a whole host of other tough diseases, everything from the flu to HIV and even cancer therapies. But hey, don't count DNA vaccines out. They are also approved and in use in humans. A key example is ZyCoV-D. That's a DNA vaccine for COVID-19 that was approved in India. And just like RNA, the technology is being tested against some really tough targets, like HPV-related cancers and tuberculosis. And you know where DNA vaccines have a really long and successful track record? Veterinary medicine. They're used to protect horses from West Nile virus, they're used as a therapy to treat melanoma in dogs, and they even protect farm salmon from viruses. Their stability is a massive advantage in these settings. So after all that, how should we really think about these two technologies? Well, there's a quote from the source material that just sums it up perfectly. DNA and RNA vaccines represent a paradigm shift in vaccinology, offering unparalleled speed, flexibility, and the ability to elicit comprehensive immune responses. They have truly changed the entire game. So the most important point to take away is this: they aren't rivals. This isn't a competition. They are two different, powerful tools for building a healthier future. One is super stable and established. The other is lightning fast and incredibly potent. Both have a critical role to play. And that kind of leaves us with one final, exciting thought. With these two incredibly powerful genetic platforms in our toolkit, the question isn't really which one is better. The real question is, what global health challenge are we going to solve next? Thanks for tuning in.